Okay, this is going to be human genetics slash heredity because heredity is a synonym for genetics. Okay, so the rules of genetics that we talked about for like all regular genetics, they all still apply for human genetics, but human genetics and heredity is going to be way more complex because we're going to have multiple chromosomes dealing with the same thing. Like let's say in a pea plant, they would only have one chromosome or like a pair of chromosomes dealing with height, but in humans, we're going to have multiple chromosomes dealing with height, and it's going to get more complex. As a side point, humans are not statistically valid because for us, like one couple can't have 100 kids. And in order for something to be tested and said a conclusion about, you need to be able to have like 100 kids. So let's say for a pea plant, we could take two parents and mate them and take 100 of their kids and then test them and say, oh, this is how pea plants are. But for humans, humans can't have 100 kids, so we can't test it. So it's not statistically valid. Anyways, that's a side point. So how do we detect human genetics? Like, how do we know what is on our genes? If you want to see if something's wrong with your genes or just whatever it is, how do we test them or detect them? So there's going to be different techniques for detecting genetics. So there's going to be screening, karyotyping, amniocentesis, and CVS. Okay, so for screening is going to be a test on some body fluid. It could either be spinal fluid, your mucus, some body fluid. And screening is not only like to look at your genetics, it's also could be to look for a certain protein, and because genetics make protein, right? So if they want to, you could look for a certain protein which chromosomes make, and if the protein isn't there, let's say, then you know something's wrong with your chromosome, a certain chromosome. Karyotyping. Karyotyping is a picture. You take a picture of a nucleus with the chromosomes, and you cut out the chromosomes, and you match up the homologous pairs from biggest to smallest. And from that, you can see if there's a chromosome mutation, because you could see if a pair ends up having three chromosomes in a pair, where it's actually, that would be a mutation in chromosome number, or if it has one in a pair, where it's supposed to have two, but it has one, that would also be a mutation in chromosome number. Or you could see if a pair are together, but one is smaller than the other pair, like the other chromosome in the pair, then you know that that there was an addition or a deletion from the chromosome. If let's say it's bigger than its pair, then obviously it had an addition. If it's smaller than its pair, then obviously it had a deletion. And that would also be, you could see if there's a mutation from looking at a karyotype. Okay. So let's say you would take an ultrasound, which is a picture of your fetus if you're pregnant. And so if you see from the ultrasound that something might be wrong with the fetus, then you can do an amniocentesis. What is that? It's a genetic test on a fetus. It could only happen in the 4th month, which is like 16 weeks. It's done using local anesthesia, and the doctor will put a needle, an empty needle, it doesn't have anything in it, through the mom's navel and through the amnion, which is the sac holding the baby. And the water that the baby is in, the water that the baby is swimming in, the baby constantly is swallowing it and spitting it. So therefore, the fluid has a bunch of the baby's cells, because when you spit out the water, it's going to have tons of cells. So the needle will take some of the water that the baby is swimming in, and it's going to take it along with the baby cells. And then the doctor will spin down the water in something, I don't know what it's called, and out will come a blob of fetal cells. And there you could either do like a karyotype or some sort of testing. But that's what an amniocentesis is. Okay, next is a chorionic villi sampling, which is a CVS. So in a bird egg, there's a chorion holding the egg together. For placental animals, the how do you say it? Umbilical cord. Basically, it wasn't there in the beginning. It used to be a chorion. So, like all the way in the beginning of a pregnancy, the baby has a chorion around the amnion. So, basically for CVS, which is a chorionic villi sampling, a needle goes through the vaginal opening, through the cervix, and it goes up, and it takes a piece of the chorion, specifically a villus of it, which is a folding, and then you could test it with a karyotype. So, yeah. But this must be done at around 5 weeks, meaning when the baby is 5 weeks old. Otherwise, there's not going to be a chorion. And that's it.